Hi, this is Sander from ImmuneWatch, and today I will show you how you can go from CellRanger into annotating the specificity of your T cell receptors using ImmuneWatch DETECT. And we'll start off by looking at the web summary of our CellRanger experiment. In this example, they measured around 9,000 cells. But if you go to the VDJ T side of things, you'll see that they ended up with an estimated number of cells of around 6,000 with a total productive VJ spanning pairs of 5,700 TCRs. You can see this here. We have the pair data, an alpha chain and a beta chain. So if you want to go and annotate the specificity of these chains of these T cell receptors, you can go ahead and browse to your output files of CellRangers, browse to the VDJ-T folder, and then look for a file called Rearrangements.tsv. I already went ahead and downloaded this file, and you can see that it's available here in my demo folder called CellRanger Rearrangement.tsv. Now, the next thing we have to do is then download the software. In this case, we will be using ImmuneWatch DETECT, which you can find here at our website, immunewatch.gitlab.io/detectdocs. We'll just go ahead and copy this specific command. To be able to download this, you need Docker installed. So, and what this will do is we'll actually pull the latest version from our website and then install it, as well as a helper script called ImmuneWatch DETECT. So you'll see here that we also have an additional script, file called ImmuneWatch DETECT. What's next is that you also need a license key to be able to run this software. You can find this by going immunewatch.com, go to the download button, try out today, and then actually request your free license key by clicking on this button. Now, once you've done that, you can easily get started by typing the command ImmuneWatch DETECT -e is the input file, in this case, our CellRanger rearrangement. -o is annotations.tsv, that's the output file. Then you have to define your license key, which I already pre-saved here, as well as saying what database you would like to use. In this case, we will be using IMWDB. All of these commands can also be found here very easily to get started. Next thing, just press enter, and you'll see that here it automatically recognizes that this is a paired chain file, meaning that you both have the alpha and beta chains available. These are linked by a column called cell ID, and ImmuneWatch DETECT will use this to actually chain together your alpha and beta chain and make one prediction for each T cell receptor. You'll see that it was fairly quick to run this, and we have then an additional file called Annotations, which we can open in any kind of software. For example, here we're opening it in Numbers from on my Mac. You can also just open this up in any kind of other application like Excel, or even in RStudio, in Python, etc., to do more downstream analysis. I'm going to delete this file. Voila, we have annotations.tsv here opened. And what you'll see is this will be exactly the same as your input file, cell ID, etc. So this is exactly what you get out of your CellRanger output, but we've added a few extra columns starting here from epitope, which shows the epitope that this TCR will probably bind to, a score, the MHC molecule, a score for the prediction of the MHC, some reference TCRs, etc., as well as the antigen name and the species name. We will go ahead and sort this file a little bit further based on the score. So we'll go from highest score to lowest score, and then we'll have a look at what antigens were found. In this case, we found some SARS-CoV-2 specific TCRs, some MART1. This is a tumor sample, by the way, specific TCRs, influenza, etc. So this is a quick demo to show you how easy it is to go from CellRanger into annotating the specificity of your T cell receptors using ImmuneWatch DETECT.